Right, question 5. Microtubules are found in all eukaryotic cells. Name the globular protein of which microtubules are composed. Bit of a gift. This is tubulin. It's actually an alpha-beta tubulin dimer, but tubulin. Name the structure from which the microtubules radiate. So that's your microtubule organizing center, your MTOC. Another gift. Honestly, you just need to know that. You also could have said that it is the centrosome. The formation and functioning of mitotic spindle fibers depends on the action of specific enzymes such as cytoplasmic dynin. The role of cytoplasmic dynin in mitosis was investigated. Preparations of pure antibodies, purified, not pure, that inhibit cytoplasmic dynin's action were injected into cultured mammalian cells at different stages of mitosis. Comparable cells were injected with a buffer solution containing no antibodies. The figure shows a cell undergoing microinjection. I don't know that we really need to do this picture, but it's obviously just to break up it a little bit. Following results were obtained. Mitosis was blocked in 73% of the cells injected with the antibody at 12 mg per centimetre cube during prophase. Injection of buffer alone had no effect on mitosis. Lower concentrations of antibodies, 6 mg per centimetre cubed, had no obvious effect on mitosis. Cells injected with antibody during metaphase or anaphase completed mitosis with no detectable differences compared to cells injected with buffer alone. Okay, state the purpose of injecting cells with buffer solution only. So this is your control that we have. So what we're doing here is to show buffer is not affecting your mitosis. Or, now being very clear, so you don't just say to compare because it's asking for this specific experiment. So you could quite happily say it's to compare the impact of buffer against when you have the actual, what we're looking at here, antibodies in. Okay. If you say control, you have to say it's the negative control, just be clear on that one. Right, valid conclusion. Right, so you look at what your data is. So mitosis was blocked in 73% of the cells injected with the antibody during prophase. That tells you that it is probably involved during prophase. So if you said that you have cytoplasmic dynin is involved in prophase, that's fair enough. You've also got that the concentration of the antibodies mattered. So that implies that there must be a level of your cytoplasmic dynin which has an effect, because if you're taking out only a small amount of it, it was still managing to function. And we also could say this bit here that it's not involved in metaphase or anaphase because it had no impact either. That's it. Okay. Once mitosis is completed, the cytoplasm separates to give two daughter cells. State the term used to describe this process. Well, this is, again, that one's a bit of a gift. Okay, cytokinesis as splitting the cell. And that's the whole question. Done.